and vice versa. Okay? So you can implement this electronically, but what you can do is control the slope of this thing, which is the gain. You can have a small change in area cause a small change in feedback, or a large change in feedback. And your medical doctor will do that with you by putting a pen of light on the edge of your eye. And the eye can start oscillating all over the place. Okay, your pupil will oscillate. Well, this is what happens as you change the gain. Here's the pupil area as a function of time. Now you change the gain and you put it higher. Here's the pupil area as a function of time. And here. So what is the difference between these situations? You can characterize them by saying, well, it looks like it's oscillating. Is this an oscillation? It's an oscillation. If actually you compute a power spectrum, you'll see a nice peak. Okay? So in fact, what's happening here is the system is going across an instability. It's actually going across what we call Hopf bifurcation. It's starting to oscillate. But there's obviously a lot of noise in this system. It's not a clean oscillation. So you can write down an equation for what's going on with this system. The simplest equation you can think of for a pupil is basically the rate of change of amplitude depends on some time constant for the amplitude to go back to some equilibrium, plus this is that feedback function that I showed you, that sigmoid. But notice that it has a delay. You need the delay, because it's there, it's 300 milliseconds. That delay is of the same size as 1 over alpha, which is also 300. So this is a situation where you need to incorporate the delay because it's on the same order of the other time scales in the system. If you don't do that, you're not going to understand the system very well. So, and it's very frustrating from a theoretical point of view because you can't do any kind of perturbation expansions. There's no small parameter. The delay, alpha, is the same size. Okay? And these are two parameters which you can actually shake around with noise and try and reproduce these kinds of oscillations. If you want more details, it's written in this paper. But basically you can show that deterministically the system goes from a fixed point to a nice regular oscillation. But stochastically, it's going across this mess of oscillations that increases in size. Okay? So this is delayed feedback 101. Yes?
Where do you put the noise?
Sorry?
Where do you put the noise?
The noise is intrinsic in the brain. In the equation, it goes into either of these two parameters, which is how much you're awake, if you're falling asleep, okay, if your level of activity of awareness changes, that's a noise, it's not constantly there. So there's noise in the motor plant and the muscles, so you can try different hypotheses in there.
Can you put white noise?
And in this case, there's a debate in this area, is the noise injected at a very high frequency, is it quasi-white, or is actually quite filtered noise? Turns out it doesn't make much difference, and therefore we can't put our finger on it. It's probably, I mean, in reality, it's just synaptic bombardment somewhere in your brainstem. It's probably pretty white, okay? So the real pupil reflex has two delays, but I showed you only one, all right? There's another feedback loop you can throw in, but that gets a bit complicated. All right, now let's go into an example where this is the neural circuitry inside a fish, okay? And this fish, they live around here. Well, actually on the other side of the Andes, they're in the Amazon, and, or in Ecuador as well. And they developed this electric sense. And the ones that I'm interested in are actually South American fish. There's a whole other collection that are in Africa, which are pulse-type fish. These are wave-type fish. So if you were to put an electrode in the water next to these guys, you'd see this nice, regular pacemaker. They're basically oscillating its current shuttling back and forth in the body at about 1,000 hertz. Okay? And it's a very, very accurate pacemaker. It's the most accurate in biology. Now, if you bring an object, so that sets up an electric field that's dipolar, you see? And when an object comes close by, this changes the field lines, changes, if it has a conductivity different from the water, it changes the field lines. If it's a conductor or an insulator relative to the water, it's going to change things. And this waveform will now get some perturbation on top of it. And the fish reads out this perturbation. It's like the auditory system. You have a carrier wave, let's say at a pure tone, and you look at the modulation. So the ear extracts the modulation. So here, the modulation goes up, and it goes up to higher brain. Okay? And this is, for example, it's going to go up into this cell, into the basal dendrites. This is the soma. There's apical dendrites. This goes to higher brain, and there's going to be feedback. So this is a classic example of this thing right here, where things go one way, but after a while they go both ways. All right? Questions so far? Just jump in with questions, as usual. Okay. So here, just to give you a better idea of what's going on, if there's an object near the fish, this is from Mark Nelson's lab, it produces a change in potential of the surface of the fish. So it's basically like vision, or like the sense of touch. Okay? The object gets closer, the change of potential, which is in microvolts here, is bigger and bigger. And this change in potential is read by receptors on the surface of the fish, and that produces changes in firing rate. Color-coded here, in spikes per second. So you can imagine a one-to-one change. You know, if the voltage goes up, the firing rate goes up relative to what it is normally. Okay? So it's a system that has evolved for millions of years, been very successful, and these fish do this kind of thing. They swim forward and they also swim backwards, and then they eat the object. They detect it, they move backwards, and they can eat it. Okay? Sometimes they'll go back and forth a few times to make sure it's something reasonable to eat. Okay, so this all takes about 1 second. Okay? Let's move down. Yeah, okay. So, sorry?
So this is from real fish?
Yeah.
Reconstruction from...
Yeah, reconstruction from a real fish. That's what Mark Nelson did. And we do simulations. We solve the relevant electrostatic field equations on a computer using finite element methods, and we can reconstruct the images, what the natural images for these fish. So this is what a metal sphere would look like for this fish. It's not something it's particularly interested in, but the experimentalists use it all the time. So a metal sphere distorts the field lines here. What is also interesting for this animal is what another fish looks like. Okay, so this is what so we can combine, you know, animals, collections of animals. So we're simulating these things. I have a student right now doing this in 3D electric field simulations to see what the natural image is as they move around. Okay, now that's an introduction. They have receptors, but we're not going to care about receptors in this talk. The receptors project up to the next stage. Okay, there's 16,000 of them, and that's it. Okay, after the receptors, however, multiple loops, enough to keep us busy, or me at least, for decades to figure out what this is.